screen right now?
Yeah, sure. Okay, hi everyone. Thanks for joining us today. This is our last and final Meet the PI series for this academic year. We saved the best for last. So today we have Dr. Marco Trizzino and Dr. Elise Fouquerel. I hope I didn't say that too hard.
Yeah, that's okay.
So they recently joined Jefferson and they're both in the biochem department. And yeah, today they're going to be sharing a little bit of what they do in their research labs, and then also talking about how they got to where they are here in their academic career. And again, I want to thank everyone who's attended all of the Meet the PI events. I hope everyone finds these useful. I know personally I find it useful to hear about the stories of all the younger PIs. So yeah, thank you, Elise and Marco, for participating and putting in the time to make your presentation. And we're all very excited to hear what you have to say.
Thank you for inviting us.
Yeah, thank you, Gina. All right, so I'm gonna start actually by telling you a little bit of my path. Or did I come, to this point? So I guess my accent can tell you that I was born in France, and so my education, I started it in Caen, where I was born, which is in Normandy. And well, I put some picture because I thought it would be nice to show you how France looks like. So Normandy, it's mostly where you have more cows and apple trees than people. It's a very nice region. So there I did, normal path, high school and everything. And I stayed after high school. I did 2 years of an institute that really shaped my training in, my bench training. So after the high school, I didn't want to go to regular university. I really wanted to have experience with the bench right away. So I did this specific school. It's kind of like an engineering school, and that gives you a 2-year diploma, which I think is for you is the equivalent of Bachelor of Science. And my major was biological and biochemical analysis. So basically I was trained to become a lab technician in a biological analysis laboratory, like process with blood work and things like that. So the thing is that after these 2 years, I thought that it was kind of routine to do this analysis, and I wanted to go further. And that's why I decided to go for
Yes,
yeah,
it's the same,
right?
in Italy.
I think it's pretty much the same in Europe. So basically, after my 2 years in this institute, I decided to go in the east of France, in a city called Nancy, to do this master. And to do this master, I had different options, and I decided to have like a specific, I couldn't really choose what I wanted as a major. So I think I was crazy enough, but I did 2 majors. And I trained in molecular and structural biology and microbiology and enzymology. And after that, so that was 3 years. And the last year was mostly lab. And I did my lab work for this master in what actually was, after that, my PhD lab in Strasbourg, which is closer to the German border. It's a very pretty city. I love this city. It's very nice. So I spent 7 years of my life there. So I finished my master in the lab where I did my PhD. And it was in the Department of Genome Integrity and Tumor Biology. And my thesis was about deciphering the roles of poly-A-deparibose glycohydrolase in DNA repair and replication. So I really started to work in DNA repair and in the poly-A-deparibose world, which is actually what I'm doing today still. And so after that, I decided to do a postdoc. And for that, I had the opportunity to go to the US. And the postdoc I did was in Pittsburgh. And I moved alone there, but my kitty came with me, and he's still around. He's an old one, but he's still here. All right, so I thought I would, show you a bit more in detail of what I did during my PhD and my 2 postdocs. So as I said, my PhD was about understanding the role of polydipyrrobose glycohydrolase, or PARG, in DNA repair and replication. During this PhD, I authored 3 papers, a book chapter, and also I did 1 year of teaching my last year of PhD, which yielded also a paper, which was a method paper, because it was the first time that PhD students were really teaching in a high school in this region of France. So it was kind of experimental. So we wrote a paper about that. And then when I moved on to my first postdoc, so indeed I did 2 postdocs, so you'll understand why. So in November 2011, it was 1 month after I graduated from my PhD. And I found this lab whose the head was Dr. Rob Sobol. And he hired me because I had experience in the polydipyrrobose world, and he had a project about polydipyrrobose polymerase in DNA repair and its link with cell metabolism. And it was good for me because it was still in the same, studying the same enzymes, but in a different context. So I learned a little bit about cell metabolism as well. So it was kind of adding another skill to my skill set. And during this postdoc, I authored a first paper in Cell Report. I had a collaborative paper, a book chapter, and a review. But then in April 2014, Dr. Sobol decided to move his lab to Alabama, in Mobile, Alabama. And although he wanted me to follow him, I didn't really want, for personal reason, and also because, well, Alabama was not really exciting for me. And so I decided to stay in Pittsburgh, and no offense to Alabama, of course. So I decided to stay in Pittsburgh, and I was fortunate enough to meet with my other postdoc mentor, Patty Opresco, who had just, again, stars aligned a little bit. So here she had just received an R1, and she was looking for a postdoc who had, skills in DNA repair field, pretty much. So we met, and we really got along really well from the first time. And what was nice is that I really appreciated her, because and my first meeting with her, she asked me, what do you want to do after your postdoc? Which was not asked to me by Rob Sobol before. And I really wanted to have my lab. And so she said, okay, so we are gonna work, and I'm gonna help you to reach this point. And it's important because I think we'll talk about it later, but having the right mentor is actually very crucial for your career. So when I started in Patty's lab, I had 2 projects. The first one was finishing a project along with a PhD student who was finishing her PhD. So I helped her wrapping up this project. It was about how UV DNA damage are repaired at telomeres. And that yielded a co-first author paper in Nature Communication and a review. And then I moved on to oxidative stress at telomere, and the role that oxidative stress, the impact that oxidative stress has on telomere integrity and also on telomerase activity. And thanks to this project, I got 2 first author papers in NSMB, and the very last one before I came here was in Mol Cell. I got 2 reviews, 1 book chapter, and 3 collaborative papers. But the big, I think, accomplishment that I had during my postdoc is that, thanks to also Patty, who pushed me a lot, is writing a K99R00. And since I had a unique background in DNA repair and PARP, and now a new training in the telomere field, I was able to link both background and come up with a new project that was really my own, and write this K99R00 that I got in 2016. And that was really the key for me to get this position here at Jefferson. I think we'll talk a bit more about it, but thanks to that, let's face it, if you have money already when you apply to a position for assistant professor, it's easier to get interviews. So as I said, I merged a little bit the PARP world and the telomere world. So I'm going to talk a little bit. So I don't know if you have, Gina, do we have questions right now, or do we leave them for later? Should I carry on? Yeah.
Yeah.
Okay. So what we do, I'm gonna just go quickly on what we do in the lab. So what are telomeres? So they are the tip of our linear chromosome. They protect them. And if you picture this car as a chromosome, the telomere are actually the thickness of this license plate. So they're very, very small, yet they're very, very important. They help, they protect chromosome from fraying. If the telomere are lost, this can happen in the cells. So you have telomere fusions and you have spaghetti-like chromosomes, and it's of course needless to say that it's very harmful for your cells. And they also have this specific structure that hide the extremity of the chromosome into a D loop. This structure is called the T loop. And this is structure that really hides the end of the chromosome and protect them from DNA damage response. So they are specifically composed of TTA GGG repeats that can reach 15 kilobases and a specific set of protein called shelterin, and also a 3' single-strand G-rich overhang that is the strand that invade the double-strand break here to the double-strand DNA to form this loop. But what's interesting about telomere is that after each cell division, they do shorten. So I could go around like with the mechanism, but it would be too long. Basically, this natural shortening in somatic cells leads to very short telomeres in some cells, and so they are unprotected and they look like double-strand breaks. And that triggers a DNA damage response, which in cells where the checkpoints are intact leads to senescence, which is basically biological aging. But the thing is that in some cells where the checkpoints are altered, like for instance, if you have a p53 mutation alongside with very short telomere, this shortening can carry on, and that leads to chromosome instability, and the cells eventually die. Unfortunately, some cells are able to re-express an enzyme that we call telomerase, and telomerase lengthen the telomeres for the cancer cells to be able to replicate indefinitely. So we are in the lab, we are really working at the border between aging and cancer. And we are particularly interested in a condition that accelerates telomere shortening, which is oxidative stress. And oxidative stress, you are exposed to it daily. It's because of pollution, cigarette smoke, the thing you eat, if it's junk food, exercises, it's everything. So it's very important to understand how oxidative stress accelerates telomere shortening and how we can overcome the impact of oxidative stress on our telomeres. And to do that specifically, we basically work on the polydipyrobose polymerase, which I told you about. I worked on it since my PhD, because those PARP enzyme, there are 17 of them, and we are mostly interested in PARP1 and PARP2. So since there are 17 members, they are really involved in a lot of processes in the cells, from cell death to chromatin remodeling, to cell cycle regulation, transcription, replication. But what we are interested in the most is DNA damage, and mostly DNA damage at telomere. And there are some more and more evidence that PARP, and especially PARP1, regulate telomerase activity and access. So this is one of the projects we have in the lab. We have also a project that studied the specific role of PARP1 and PARP2 in repairing oxidative stress at telomere. And we also have another project about replication and the role of PARP in replicating telomeres, because their sequence TTA GGG is very particular and very hard to replicate, and that leads to replication stress. So this is the main project that we have, but I wanted to focus a little bit more in the project that's related to oxidative damage, because we have a really specific tools in the lab that no one else has, beside my postdoc mentor Patty, that I developed when I was a postdoc, that we call the fluorogen activating peptide, or FAP system. So it's a chemoptogenetic tool, and for that we have this homemade red LED. So how it works, we have this FAP peptide that's fused to a sheltering protein, TRF1, which is a protein that's specifically expressed at telomere. So basically we can only express this FAP system locally at telomere. And the small peptide, the FAP, is able to bind a dye that we just incubate with the media. That's a green dye that we call MG2I. And when it's bound to the FAP peptide and exposed to this red light, it gets excited and it releases a singlet oxygen, which reacts with the DNA to induce ethoxoguanine. So it oxidizes basically the guanine. And as I told you at the beginning, telomeres are composed of TTA-GGG. So it's 50% of Gs. So basically we are able to induce a lot of ethoxoguanine at telomere. And this tool that we use, this is really the base of our project in the lab. So we also have other techniques that we use. So mostly that's kind of specific to telomere. So we do a lot of thousand blot, and those they allow us to check at telomere size in bulk in cells. We do a lot of microscopy. So we have our own fluorescent microscope. We do a lot of like regular biochemistry experiment, and that's the tool. And finally, we also do telomerase activity assays. They are very tricky. And also that's a whole picture of Caroline. So also, of course, when you start in a lab that's just starting, you have sometimes IKEA tools to build. So it was actually a funny picture to take. So that's basically pretty much it for my lab. And so Gina also asked us to talk about how and why we came up, like how and why we chose this particular path. So I thought it's a lot of writing, but I thought I would walk you through a little bit and we can discuss more if you have more questions. So my path in academia, the whys. So why did I decide to do a PhD? So as I told you, my first training was in this technology school in which I was really trained on the bench, but to do kind of the same analysis. So I really wanted to see something else off the bench and, work from the routine because I like the challenge and also because I could. So let's face it, fresh education is free. So it was easier for me to really go further. I had the motivation, of course, that's the most important. French PhD student salary is very good. And I also obtained a grant from the Ministry of Higher Education for 3 years, which is the process, of course, and I had the grades to go further. So after that, why a postdoc? So I wanted to explain my knowledge and my skill set, which I think was very important to start my own lab. Of course, I loved research and I didn't really want to go to industry. I was not really attracted by this world. So that's why I thought, I'm gonna stay in academia. And why abroad? So I could have chosen another country, but I just met my postdoc mentor at a conference for Observable, and he was in the US, and we liked each other. So this is why I decided to go to the US. But also, I think it was very interesting for me to be exposed to a new system of research because US research is very different from the French system. Of course, I was really excited to meet new people with various backgrounds and experiences. And I think US is very unique in that sense because there is so many different nationalities of people you can meet, and I think that was the most interesting and exciting for me. And I also wanted to increase my chances of finding an independent position. Of course, if you go abroad and you meet more people, you have also more chances to develop your network. And also, not a little thing, it's also was good is to improve my English, because at that time my English was very bad. Well, I didn't lose my accent though, but it's fine. All right, so also I wanted to just talk a little bit about applying. So the process of applying to a postdoc, I'm sure this is things you know already, but I wanted to talk about my experience. So of course you can spontaneously apply to labs. For me, I did it to 2 labs and it didn't work. And what worked for me is by going to an international meeting, and when I was presenting my poster, some people came and I started to say, well, I'm going to defend my PhD very soon. So I'm looking for a postdoc. Do you have a position? And this is how I found, and I met Dr. Sobol, and this is how I found my postdoc actually. And of course, you have the classic answer to an ad, which worked for Daniela, who's my postdoc actually. She came to my lab by answering to my ad. So how to apply? So for you, it's still, you still have a long way to go for that, but I think it's interesting to see the process. So how to apply to an assistant professor position, of course, when it is the time. You may have understood that you need quite a bit of papers and a bit of experience in your lab. You have to, what's important, I think, it's to be open with your postdoc mentor, because you have to avoid conflict, because of course you're trained in this mentor lab. So basically you have to agree with your mentor with what you can take with you in your own lab and leave and not take with you. So that's very important. And your postdoc mentor, you need this person for a while, even when you start to be independent. So I cannot count how many letters of recommendation I needed my last postdoc mentor to write for me to get a grant. And also it's also true for the PhD mentor. I'm still sometimes asking letter of recommendation to my PhD mentor. And I would recommend to start at least 1.5 years before the end of the postdoc, because it's very long. It's a full-time job. It's very stressful. You have to handle, of course, your experiments, as a postdoc, finishing your papers. But you also have to interview, you travel a lot. Well, with the COVID time, it's not the case, but I'm guessing that when it will be time for you, you'll be able to travel a lot. Send tailored application. I think it doesn't have to be told, it's normal. You have to send enough application, but in my experience, don't send too many, because I heard a lot of my colleagues who got too many interviews to handle. And maybe also too many offers that it was very hard to choose from. And also it's very hard to say no to people that you meet face-to-face. And also keep track of your application, but that can also be applied to when you apply to postdoc, it's the same. Make sure that, you can resend an email and say, did you see my email? I'm really motivated. What's your answer? So keep track of that. And then you have to prepare with colleagues for your interview. So that's the same for postdoc, because although I didn't get a regular interview to get my postdoc position in the US, I do believe that postdoc interview are the same as assistant professor interview. So you meet with colleagues, you go to the university, you give a seminar. So it's very important to prepare that with colleagues and have feedback. And particularly with the assistant professorship, you have a chalk talk to give, and this is a really tough part of the interview, and that's very important to prepare and rehearse with people who already have the experience of chalk talks. And then if you get multiple interviews, which is good, try to maybe settle for a schedule in which you have the one you are the least interested first, so then you can get practice. And then, of course, you have to study your itinerary carefully, read a couple of papers for each person you will meet, because, of course, you have to have a smooth conversation with them. So it's important too. And then something that really helped me in the process of applying for an assistant professorship is this book. So it's reedited every year, and I would recommend it to you. It might also be useful actually for applying to postdocs as well. And finally, the last slide is how did my training prepare me for leading a lab? I think it was quite unique because I did 2 postdocs. I know two postdocs might sound a bit rough, but actually for me it was actually very positive, because I could, gain another skill in the telomere, like more knowledge, and that's what helped me really put this K99R00 together, because it was pretty unique. It was the first time that someone were merging the telomere and the PARP field together. And I think also something that I'm not really comfortable is giving talks actually at meetings. It's not really my, I don't really enjoy it. But if you're a person who likes it, it's good. But my PhD and my postdoc, I really push myself to present poster, to give talks, ask my mentor if I can apply, because I think you have to, step out of your comfort zone, and this is how makes you better at, presenting your data. Also, this postdoc, the two postdoc gave me also experience beforehand in mentoring students. But of course, it's not the same level of involvement, because when you're a postdoc and you mentor a student, it's for your mentor. So it's not really, it's less involvement. But when you are an assistant professor, you want your student to really succeed because it's good for you. So I think it's a bit different. It's more challenging, but it's fun. And then, I think I got two different experiences because I had two different mentors. So my first mentor was really hands-off. It was basically not in the lab. So I was very, very independent from the beginning. So that helped me actually a little bit. It was annoying sometimes, but it was also really helpful. And at the same time, my second postdoc, although she was hands-off and she was not micromanaging, she was more like willing to share her experience as a mentor. And I think that was very useful for me, because from those two type of mentoring, I could really take what I wanted to export to my lab and really see what I didn't want to apply in my lab. So I think that was very helpful. And then I do think, I just put also that, of course, there is still a lot of improvisation, because you don't know, the people you're going to hire, you have to adapt, you want them to succeed, it's very important. But of course, you do mistakes, and you have to be ready for that. And this is what it is. And you have to be patient. That's something I was lacking a lot. And I think I grew more in that. I'm still on the learning curve, but it's progress. And do not hesitate to ask for help at any stage of your career. Just ask, because people don't come like that. You have to ask. But when you ask, usually my experience shows that they're actually really willing to help you. You have just to express it. And of course, makes sense. Work hard, but make room to unwind a little bit. Friends, family, whatever you like to unwind. I think it's important at every stage of your career. I guess that's it for me. I don't know, I was quite fast, but if you have any question. All right.
Thank you, Elise. I appreciate your advice, especially at the end. Does anyone have any questions specifically pertaining to her research, perhaps? If not, we can save all of the career development for the end. No. Okay, Marco, you're up.
Okay, thank you. Let me just share the screen. Okay. Can you see my slides?
Yeah.
Okay, so I went less into detail about the research we do, but I still have a few slides about it. But if you want to talk more about it, of course, I'm here. And I focus a little bit more on my path to get this job and to get this position. And I already, can tell that I share most of actually all of the advice that Elise just gave in her presentation. And so what do we do in my lab? So I joined Jefferson 18 months ago, a little bit more than 18 months ago in 2019. And so what are we interested in? So you know that eukaryotic cells are made of hundreds of different cell types sharing the same genome, but still having different functions and shape. And I've always been attracted by, how is that possible? How is that possible that with the same genome, you can make a blood cell or a neuron or a bone cell or so on? That's because, we have 30,000 genes, roughly, give or take, in human cells, but not all of them function at the same time. So we're very interested in how gene expression is regulated in a context-specific fashion. And how does that work? So you have your gene here, and then you have several non-coding regulatory elements in the genome that could be distal, such as enhancers that are found several kbs away, or proximal, such as promoters that are close to the gene. These are called cis-regulatory elements. These elements, they need to crosstalk to each other, normally through looping of chromatin, in which enhancers and promoters get in touch and close to each other. And this activity is regulated by transcription factors, by protein complexes, chromatin remodelers, and so on, to ultimately recruit RNA polymerase II at the promoter of the gene, and to start the transcription of the RNA and the expression of the gene. So in my lab, we are very broadly interested in all of these actors, the cis-regulatory elements, the transcription factors, the protein complexes, POL2. And specifically, we have two major research areas going on in the lab. One is to investigate how gene expression is finally regulated in cell-fate determination during development. So you start from a stem cell, you need to make a neuron, or you start from your embryo, you need to make your limbs. Anyway, you still need to express genes, specific genes at specific time points. And when these genes don't work well, you get developmental disorders, you get syndromes. So for example, we are interested in genes whose mutations cause developmental syndromes. And how do we study them? The main approach we use is to use stem cells, and specifically those that are called induced pluripotent stem cells, that are artificially reprogrammed stem cells that you can reprogram from any cell of your body. If you treat it with the correct types of transcription factor, you basically treat them with a cocktail of pluripotency factors. In a matter of weeks, they will go back to stemness. They will become stem cells. And so what we do is to get cells from patients, kids mostly, with developmental disorders, with mutations in specific genes. We get skin fibroblasts. We make stem cells back from the fibroblasts. And then we differentiate them, for example, into neurons or into neural crest cells to see why the mutation of these genes are so important in these developmental pathways, and specifically, what are the molecular functions that are impaired when the genes are mutated. And so Luca, my first postdoc, is working on this kind of projects. And then the other second major areas we're interested in is evolutionary biology, and specifically how this developmental gene expression that we just discussed has evolved in humans and in primates more in general, and specifically the role that transposable elements had in this evolution of developmental gene expression. You may or may not know it, but transposable elements are these jumping genes, these parasitic DNA elements, account for over 50% of the human genome. Human genome is huge, but half of it is made of transposable elements. And these elements, they're parasites, but sometimes the genome starts a sort of a mutualistic relationship between these elements and the genome, and starts exploiting them, using them for its own advantage. For example, by recruiting them into gene regulatory networks, as they provide a substrate for binding sites of transcription factors. So a specific transposable element might be particularly interested in a specific cell type because it has binding sites for master regulators of that specific tissue and cell type. And to study that, we still use stem cells. For example, we compare evolution of gene regulation in human and chimpanzee. So we have stem cells of humans, stem cells of chimpanzee. My postdoc Shruti Paturi is working on this, and specifically we are interested in the evolution of the hippocampus, because in the hippocampus many transposons are active, and these transposons are not active in other regions of the brain. So we're trying to understand, why do you have active transposons in the hippocampus? What's their role in the hippocampal gene regulation? And to study that, we are specifically focusing on a family of transposable elements that are human specific. So you don't even find them in chimpanzee. And by comparing human with chimpanzee, you'll be able to see why these transposons are active in humans and what's their role in the generation of the hippocampal cells. And so my postdoc Shruti works on this. And then I have two more postdocs, because I was lucky enough to get a big NIH grant and at the beginning, and a foundation grant, and two more postdocs. So Daniela, another Daniela, she's from Colombia in this case. is working on the role of transposable elements in the expression of genes associated with aging. She's interested in aging and across mammals in this case. So she's studying transposable elements in the genome of multiple mammal species. She's a computational biologist, so her approach is entirely computational in this case. And then finally, Chiara, my 4th postdoc, is working on trying to understand why transposable elements tend to be derepressed and activated during onset of Alzheimer's disease. So we got stem cells from Alzheimer's patients. We're making hippocampal neurons, even in this case, and we try to compare this sort of differentiation with cells from healthy donors, and we try to see why these transposons are active in the Alzheimer's disease and not in the healthy patients, in the healthy donors. So this is just to give a broad overview of what we do in the lab. But I'm generally, open in everything that is associated with regulation of gene expression, with a mostly focus in development and evolution. Now, how did I get here? Right, what was my path to come here? So I'm from Italy. I'm from Milan. Milan is in the north of Italy. So after high school, I went to this university. It's a state university. It's not a fancy one. It's probably the ugliest campus in Italy that you can find. It's really an ugly place. The sky is always gray. It's a horrible place. But it's a good university. And there I majored in biological sciences. I got my bachelor there in 2005. And as Elise said, in Italy, if you want to do a PhD, you need to get a master's first. And that's why, since I already knew that I wanted to try to work, to have a career in research, I enrolled in a master in evolutionary biology in Rome at La Sapienza University, which is one of the, so unlike this ugly place, is one of the most beautiful places in which you can study, most beautiful city in the world, one of the most ancient universities in the world, very prestigious, a great place, great public university. I did my master's degree and my PhD in the same department of biology and biotechnology at Sapienza. So my master was between 2005, 2007, and then my PhD between 2007, 2011. So I realized when Elise presented that we got the PhD in the same year. And that's quite a coincidence. And so from this year, we're both no longer new investigators for NIH, right? After 10 years, if I remember correctly. Well, early stage investigator, at least.
Yes, that's sad, but that's true. Yeah.
Yes. So yes, my PhD. So what was my PhD about? So it was not about what I do now. So I unlike Elise, was pretty consistent in her path, working on DNA repair and PARP and telomeres. I did my master and PhD on molecular evolution and population genetics in insects. So for example, I studied the RNA, RNA, you probably know, they not only have a sequence, but they also have secondary structures. So they fold in specific ways. So what I studied as a master student and PhD student was how this RNA folding varies from species to species in insects. And if you can use that to do some, broad level population genetics to characterize, to see if there are like haplotypes of RNA secondary structure, is something that no one did before. And so I ended up finishing my PhD with 9 first order publications in, not super prestigious journals, a couple of good ones, but, nothing major because, the field is not a field, it's not really a biomedical field. So it's not very easy to publish in journals, with a huge impact factor. But still, I had 9 first order publications in 7 years. And, a couple also collaborative papers here and there. And then in 2011, I graduated, but I also got married a couple of years earlier in 2009. And my wife had a great job offer in Milan. So we decided to move back to Milan. And once we moved to Milan, I couldn't find a postdoc position right away because finding a postdoc position in Italy is pretty tough, which is why for 2 years, 2012 and 13, I was out of academia and I worked for a project manager as a non-government with a non-governative organization, an NGO that was working in working projects on writing research projects mostly for Africa to work with, Africa biodiversity, to for endangered species and environments. It was actually a very nice experience. Unfortunately, didn't get to go to Africa, but it was great to work. It felt like very rewarding for me. But during these 2 years, even if I technically left academia, I kept writing papers and publish with my PhD advisors. As Elise said, it's very important to keep a good relationship with your advisors at every level. But then I really knew I wanted to go back to academia. I really knew I want to go back to do a postdoc. And since in Italy things were really tough, we were still in the middle of the economic crisis, and my wife was actually not super excited about her job that moved us to Milan. I decided to look for positions abroad, and specifically mostly in the United States. Like Elise said, United States is where the money to do research is where the best research is done. So I was really attracted by this kind of environment. So I applied to a bunch of positions. I actually got more than one offer. I got an offer at University of Kansas in Lawrence. I got an offer at Syracuse University, but then also I got an offer at Penn, which seemed like the best one to go. And so in 2014, I moved to Philadelphia with my wife and also my cat, like Elise. And I went to Penn, Department of Genetics, in the lab of Casey Brown. And there I worked in still evolution, but in this case evolutionary genomics, and specifically here is when I started to be interested in transposable elements and the role that they have in gene regulation and how they shape basically new cis-regulatory elements in the primate genome. So new enhancers, new promoters, how are they generated? They're made from scratch? No, they mostly come from transposable elements that are recycled by the genome. And so we studied that in primates, and we published a couple of nice papers, one in Genome Research, which is one of the leading journals in the field of genomics, one in BNC Genomics, which is also a good journal. And this was in 2 years. I did, my boss had a grant for 2 years, so he could pay me for 2 years. But then I won, in 2016, when my position at Penn was finished, I published my papers. I felt like I need a little bit more training before trying to get a faculty position. That's why I applied for other postdocs, and in this case I found a position just across the street in the gene expression regulation program of the Wistar Institute, which is a cancer center that is found like in the middle of Penn. It's in the heart of Penn campus. It's a really beautiful place. And in this case I was working in the laboratory of Dr. Alessandro Gardini, another Italian like me, and we worked in developmental genomics and gene regulation, which is basically part one of my research interest that I showed before. We studied how an enhancer get activated during, for example, hematopoietic development. So when you make blood cells, how do the enhancers get activated properly? And we published this paper on Molecular Cell and another paper on Cell Reports. These are two very good journals as well. And then I published a third paper basically right when I started here. It was actually accepted not long ago, on Science Advances, also on the same topic of enhancer regulation during hematopoietic stem cell differentiation. So 3 more papers, so 5 postdoc papers total: Genome Research, BNC Genomics, Molecular Cell, Science Advances, all good journals. But I didn't have the big one. I didn't have the Nature paper, the Science paper, the Cell paper. Like, I got very good papers. Molecular Cell is great, it's the best for molecular biology. Genome Research is the best for genomics. But when you're a postdoc, everyone tells you you really need a Cell paper, a Science paper, a Nature paper to get a faculty position, or it's going to be very tough for you to get interviews. So the other option was you have to get a grant. So like Elise said, she had a K99R00, which is a grant that basically gives you money to start your own lab. I tried twice. First time the grant was discussed, but with a pretty bad score. I resubmitted. The score was really good, but was like 1 or 2 points right below the pay line. So that sucked. I didn't get it. And it was too late for me to submit again because you have eligibility until you're, the end of your 4th year of postdoc. So I had just to try and go to the job market without a Cell paper, without a Nature paper, with a Science paper, and also without a grant. So, and another thing that people tell you is it's very important to have a very, a senior, very well-known postdoc advisor to get interviews for faculty position. And it turns out that both of my postdoc advisors were new assistant professors like me right now. So technically it seemed that I did everything wrong, right? I didn't get the senior advisor, I didn't get the grant, and I got many papers, but I didn't get like the big paper. So what were my chances? They told me like, you have to apply a lot, but you may still do it. And that's what I did. Unlike Elise, I did many applications. I did some 60 applications in the US and some 20 applications in Europe. And I got a fair amount of interviews in the US, like 5 phone interviews, which is a very preliminary stage of the interview, and 2 on-campus invitations in the US. Like 2 out of 6 is not really well. It's not really good actually, but I still got them. It's very hard to get a campus interview. And these interviews were at Jefferson here and Wayne State University in Michigan, a good state university in Michigan. And then since I'm European, I said, let's try also to go back to Europe. I applied to some 20 places. I was more successful there, especially in the UK, in which I got 5 on-campus interview. So basically 1 out of 4 of my application in Europe resulted in an interview. But in that case, I got only 1 offer at the University of Kent, which is in Canterbury, near London. It's like 45 minutes outside London. So very nice place, beautiful place, but the offers was really not competitive money-wise with the ones I got from the United States, Jefferson and Wayne State. So I ultimately decided to accept Jefferson. And so far, it's been working really well because, for example, my K-99 that didn't get funded, I basically recycled it for a foundation in the very first few months I was here, and I got funded with a lot of money. And then I got my big NIH grant, 5-year grant, a few months later. So I got really lucky, and I have now a lab full of people and nice science we do. A couple of advices here too, like Elise did. In my experience, diversifying the training was really important because, for example, I came from a genetic background when I was a postdoc, when I was a PhD student, and then I transitioned to, computational genomics, mostly computational genomics, and I learned a lot about computational genomics in my postdoc, my first postdoc. But then my second postdoc is where I really learned a lot. I did a lot of bench when I was a PhD, but it was not like the most updated bench, right? I learned a lot of cutting edge genomic techniques, molecular biology, biochemistry, a lot of everything in my second postdoc in the 3 years I did at Worcester. And so when I went to the job market, I had this nice mix of computational and wet bench skills that really help now because in the lab we can do a little bit of everything. And so diversifying is important. Trying to get a postdoc, it doesn't have to be something completely different, but to get a postdoc in a field that is a little different than what you did your PhD, or at least in a place in which you can at least get new skills. And then, like Elise said, I agree, doing 2 postdocs is not bad. It's actually good if you can be productive in both. That's actually going to be in your favor. And when you're a postdoc, especially, try to do, Elise basically said all of it, but try to do talks, to get talks at conferences. It's very important to get noticed by people in the field and to network. My 2 interviews in the US, I have to be honest, Jefferson and Wayne State, I have some sort of connections I made in both places, right? So that most likely counted. In the UK, I have to say, I did 5 interviews in 5 different places in which I had 0 connections. So the UK seems to be really completely like meritocratic, like you have a good CV, you get an interview. That is great, actually. The problem is that they don't have money. The salary they offered me as a PI was lower than my postdoc salary in the US. So it was not really convenient. I had a baby coming on the way, a wife to and the family and everything. So it really didn't seem convenient. Be persistent and, don't worry, even if you don't get the big national paper, science paper, if you can get like Elise, Elise got molecular cell like me, she got NSMB, Nature Communications, like again, molecular cell, Science Advance. These are all very good journals, even if they're not like the top journal. If you can see that you consistently produce in top journals, in good paper, in the good journals, this is gonna be viewed very favorably. And it's very important to practice every step of your interview, especially something that I did not consider and I failed miserably, the first 2 I did, was the phone interview. Phone interviews are really awkward and really hard because you don't see the people. The people make you standardized questions that the interviewers, they make standardized questions that they make to everyone, every candidate they're interviewing. and they do not make any comment on your answers, and they do not stop you. So you can, what I did the first interview, I just kept talking and ranting, and it was horrible and very awkward, and no one stopped me, and I lost track of what I was saying. It was at Rutgers, and never forget, it went really horribly. But then, once you practice and you do a couple of these, it goes better. But it's really hard, and people don't tell you that the phone interview is so hard. And the other step is the chalk talk. When you get on campus, you get to give a seminar on your research, and that's easy because that's something you've done a lot. But then you have to be, come for a second day. You go to a board with a chalk, and you have to present basically your first grant, and that's hard because you don't have slides, and you need to convince people that your research is fundable. And get to practice this because it's gonna be helpful, because you don't see any of this before you go into the job market, so you really don't know how that works. So that really helped me practicing this. I asked my postdoc mentor to organize one of these with the department faculty, like a mock chalk talk. They butchered me. It went horribly, but it really helped me understand what I was supposed to do. And that's why I think I got 2 offers out of 2 interviews in the US, because I did 2 really good chalk talks. I assume, I don't know. And that's it for me. And like I said, why did I want to do a postdoc? Why did I want to do a PhD? I have to say, I knew almost immediately when I was an undergraduate that I really want to have a career in academia and try to do research. I like, the time spent as an undergraduate in different labs, just walking around the labs, see people, doing research. That really attracted me immediately. And that's why also I wanted to do a postdoc, of course. And why the US? Because US is where the funding is. There is no country in the world that is competitive, funding-wise competitive with the US. And so that's why we came. There are so many more opportunities than everywhere else. And it's not just about the money, it's about the approach, I think, in my opinion. And that's it. I'm here to take questions. I only have a few minutes actually, because then we have class at 4, like Gina. But 5-10 minutes, I guess we can get.
I have a question.
Yes.
Sorry, I'm on the console call, which is why I start. How different can you go in a postdoc? I'm in like a neurodegeneration lab, and I've always been kind of interested in neurodevelopment. And if like something came up, like is that like, obviously I think it would be hard to go from like neuro to like strict biochem, but like, I'm just wondering, like both of you said to diversify your postdoc training, and I'm wondering like how far away is too far? Well,
you want to go first or?
Sure. I mean, my example is that there is not too far. I went from INSET, molecular evolution and genetics, to primate gene regulation and human developmental genomics. So there's really, as long as you can be motivated and be productive, and then when you go on the job market, explain what your vision is, that you have a vision that is stemming from your postdoc work, of course, but at the same time is different than what your postdoc mentor is doing, then there is not really like, there's not too different, too much. You can go everywhere if you're motivated. It's really up to you, in my opinion, but Elise might say something completely different.
I think I know, well, so technique wise, maybe there are stuff that in some lab that, there are some training that you require beforehand, but when you go to a lab that has a different topic, but uses the same tools, at the end, you can adapt to anything, right? You're able to work on the bench. So even if it's different techniques, but you know how to pipette, you know how to do cell culture, you can adapt to any other protocols, I believe. So I think topic-wise, you can be very different.
Thanks. Marco, you're still sharing your screen, just FYI. All right, thank you both.
Yeah, I actually had the same question, because I know, like, Elise, it seems like your postdocs, in terms of your PhD training, like I know other PIs that were on the seminars, they talk about having like a niche, like, so you stuck within the same field, where Marco, you like completely, I guess, diversified what you did in your postdoc training. So
for me, it was still very different because the telomere world is extremely precise. Like
yeah,
it's
exactly.
it's very different. I had to learn a lot, you know, and catch up a lot of literature because it's very specific. So it was not very, it was DNA repair, but it was still very different.
Different, yeah. So would you say, I mean, did you benefit a lot from kind of like stepping out of your comfort zone in terms?
Sure.
Yeah.
Yeah, for me too. And of course, you know, the very first few months of my first postdoc, the learning curve was very steep because it really didn't have any background on what I was doing. But, you know, it was really important and it worked out.
Mm-hmm. Okay. Does anyone else have any questions? Because I know all biochem people, we have journal club in 2 minutes.
I'll ask a quick question really quick. Did you guys feel prepared to mentor PhD students, or did you feel like that was a hard adjustment to get used to? Because I feel like, I don't know, being mentored is different than mentoring, if that makes sense. So just curious.
Yeah, I think for me it was, I mean, maybe Natalie can tell you, but it's sometimes difficult, yes. But not only PhD students, I think it's because you have different personalities, right? So you have to adapt and you have your own struggle too. You're human as well, right? So it might be difficult sometimes to, but again, I think you have to keep communication open. I think this is very important. Not, you know, if you have to tell something, just say it. Don't be an asshole. But I think it's good to, you know, it's good to talk openly because you want your student to succeed, right? So, but I may have done mistakes, and I know I've done. So, but it's a learning curve, and I think you have to warn when you're a young scientist. You have to warn your people, the first people you mentor, that there might be some bumps on the way. But you know, so then they are ready.
Yeah, and I agree. Because of getting these grants pretty soon, I ended up having a pretty big lab basically in my first year as a faculty with 4 postdocs. I have 2 rotation students right now, a master student, a technician, and it's quite overwhelming. And I'm definitely still in the learning process. So it was probably, this time would probably be better to have less people and adjust, but I guess it is what it is. And they tell you, write the grants, get the money, and that's what happened. So definitely I have to learn a lot still. And it's probably never gonna end, the learning, you know.
Yeah, cool, thank you.
All right, well, thank you, Elise and Marco. I really appreciated your presentations.
Thank you. If you have any other questions, don't feel free to email us.
Yeah, same, you know, even more informally if you have any question about everything.
Yeah, no problem.
Okay, thank you.
Thanks, Gina.
Everyone have a good day.
You too.